In the interest of time, I think we'll move right on to Dr. Junhao Wen's pre-recorded talk. I'm sorry we haven't been able to get to all of the online questions, but can I suggest please come up to the front after the session. Speakers can hang around for 5 to 10 minutes at the front, and feel free to approach any of the speakers with your questions and comments and so on. So without further ado, we'll move on to our final speaker, Dr. Junhao Wen, who will be presenting a pre-recorded talk.
Hi everyone, I'm Junhao Wen, an assistant professor of the Laboratory of AI and Biomedical Science at Columbia University. And the topic of my talk today is integrative analysis of aging and brain diseases using multi-organ and multi-omics biomedical data. I have no disclosures. As a brain imager and machine learner by training, if I would like to summarize my scientific interest into one sentence, I would say AI and machine learning can see what we don't see as a human being. That being said, I'm not claiming at all that machine learning and AI can totally replace the critical role of clinician, like neurologist, radiologist, etc. Rather, I would say AI and machine learning methodology should help or complement the insights from the clinician that benefits patients. And a critical question here, given the fact that there are more and more rich biomedical data available to the brain imaging field, how we can efficiently integrate those AI machine learning methodology on top of those multi-scale, multi-model biomedical data. And I think to better understand those data and to perform integrative analysis on top of those AI and machine learning methods, which can largely benefit the research community, but also the patients and clinicians. In my personal opinions, there are several perspectives and keywords that I want to highlight. The first one is multi-omics. Like I mentioned, as a community, we have largely analyzed brain MRI, neuroimaging data to understand the human brain and also brain disorder like Alzheimer's disease. But more and more, I have been very interested in bringing additional omics data, including genetics, genomics, proteomics, and even in the future at single cell resolutions. And of course, the second key part of the research is, here in the lab is to develop and also apply those AI and machine learning methods, including conventional statistical methods, conventional machine learning model like linear SVM, but also include more cutting edge deep learning models. And this can be applied to different machine learning tasks. For example, brain age prediction in a regression model and disease classification using some neural networks to classify Alzheimer's disease patient at an individual level. Another example could be, can we use AI and machine learning to find those disease subtypes, which is a clustering problem? And hopefully those AI and machine learning methods can benefit downstream clinical tasks like drug development, patient stratification for personalized medicine, etc. And finally, another key aspect that I want to highlight is that to leverage those rich biomedical data, model human aging and disease at multiple multiple organ system, which we refer to as multi-organ approaches. For the subsequent presentation, I would like to highlight several studies from our group on how to integratively analyze those rich biomedical data beyond brain MRI to better our more holistically understand human aging and diseases. And in a recent publication from last year, we have used multi-model brain MRI data from the UK Biobank, including T1 weighted MRI, diffusion MRI, especially for the DTI matrix, and also resting state functional MRI for the functional connectivity, and developed the three multi-model brain biological age gap, what we call brain BAG. And using those three BAG as phenotype, we linked those BAG to underlying common genetic variants using genome-wide associations. And of course, the GWAS only tells you association. It does not validate any underlying biology. And to do that, I'm going to show several different post-GWAS analysis to partially validate those genetic signals. On the figure on the right side basically show that the genomic loci underpinning the three different modality BAG does not overlap with each other, which might indicate they reflect different underlying biology. And to answer that question, we performed the phenome-wide association, basically link the genetic variants, significant SNPs, back to the phenotypic field in the GWAS literature, and we confirmed largely those genetic variants linked to each specific BAG corresponding to their original phenotypic domain. For example, the genomic loci linked to the gray matter BAG largely linked back to the gray matter volumetric matters. And another approach we could potentially validate those genetic signals is to understand those potential causal relationship between those what we call AI-derived phenotypic biomarker like the BAG here, and also with some other disease endpoints, some other imaging-based biomarkers, etc. In the follow-up study that we published last year, we extend the biological age gap, the brain BAG, to multiple organ systems, in total 9 different human organ systems, including cardiovascular, metabolomic system, etc., etc. And here, just to specific example how we can dissect the statistical association towards potential causal relationship. The figure on the left side, basically we have used the Bayesian colocalization methods, try to understand a specific at the specific SNP level, whether two imaging clinical traits or imaging phenotypes could exert potential same causal effects on the same potential causal SNPs. Here specifically, we use a pulmonary BAG developed from clinical traits related to the pulmonary system, and perform the colocalization analysis with one of the underlying pulmonary features. And as we can see here, one specific SNP on the chroma 4, which showed a very high statistical causal signal between these two traits. However, Bayesian colocalization method only tell you at the local level, like a specific SNP, whether this specific SNP exert potential causal relationship on two clinical traits. And we can also use Mendelian randomization to look at the global effects at the trait level. For example, the figure on the right side, basically we are testing the potential causal relationship between the non-organ specific BAG beyond the brain and different disease endpoints like Alzheimer's disease, sleep duration, and body weight, and type 2 diabetes. For example, one example here is we found Alzheimer's disease is positively causally affecting the brain age gap, BAG, and the brain age gap will be negatively associated with your sleep duration. In 2 follow-up papers we published this year, we have extended this biological aging framework towards a multi-omics data. Specifically, we developed 5 plasma metabolome-based BAG and also 11 proteome-based BAG. Try to extend this framework towards multi-omics data to represent organ-specific aging trajectories. And in these 2 papers, I just want to highlight another critical question in the biological aging clock field. What is the goal of developing such an age prediction model? I would argue that it's not to achieve a better model performance, that is a lower mean absolute error or a higher Pearson correlation R between the predict age versus your calendar or chronological age. Rather, I would say the goal here is to provide an aging biomarker that can predict cross-domain clinical phenotypes, for example, cognition, mortality, or disease status. In the figure on the right side, basically, we have demonstrated that using the brain-specific proteome-based BAG, we showed that if we derive those brain PAG at different, like from the under-fitted neural network versus medium-fitted neural network, and lastly, overfitted models, and we can show an inverse U-shape. Basically, that means a mediumly fit neural network which achieves a medium mean absolute error in our observation is going to be 5 years old or around 5 years old, actually provide the highest prediction power to predict cognition, mortality, and disease status. I think that is a very clear message that we definitely don't want to overfit our age prediction model itself. Rather, we want to have a biomarker that can be more predictive for cross-domain prediction. In another study we published this year, we have shown how to integrate brain MRI data and genetics, try to understand the disease heterogeneity across 4 brain disorders. In this paper, we found a large imaging pattern overlap between those disease subtypes across 4 brain disorders, specifically for the second subtype of autism spectrum disorder and the first subtype of schizophrenia. And in addition, we found a high positive correlation between those 2 dimensions or subtypes. And using like genetic analysis, specifically Bayesian colocalization analysis, we have found actually one potential genetic SNP that can exert causal inference simultaneously to the 2 imaging dimensions. And that's one specific example how we can provide genetic insights to support those imaging overlap between the 2 subtypes. Going beyond imaging genetics, in a follow-up paper we published this year, we showed how to integrate additional omics data on top of imaging genetics. Specifically here, we integrate like plasma proteomics data from the UK Biobank into brain imaging genetics to establish the brain-heart-eye axis. And by integrating those plasma proteomics data, we could provide specific organ, cross-organ interconnections that can be evidenced at the proteomics level. Finally, I also would like to highlight our team has been practitioners in open science, and we have made our resources code GWAS summary statistic publicly available at the medicine web portal. Finally, I would like to thank all my collaborators who made this research possible, and thank you for your attention. I would be very happy to answer any question.
So that brings us to the end of the session. I'd like to once again thank our stellar lineup of speakers. I think it's been a really informative session. Once again, if you do have any questions, please feel free to come up and speak to any of the chat with any of our speakers. Unfortunately, we haven't been able to go through all of the online posts and questions. So thanks again.